I'd like to tell you a little bit about malaria in case you don't know. Malaria is considered the greatest killer of mankind. In 2019 alone, the world experienced 229 million cases of malaria. Over 409,000 people, mostly young children and pregnant women, died of this preventable and treatable disease. 94% of those cases and deaths were in sub-Saharan Africa, where our work takes place. Malaria Partners International is a nonprofit organization founded and run by Rotarians. We are working to ignite the international campaign among Rotarians for the global eradication of malaria. We advocate for support of malaria elimination through presentations at Rotary clubs and district conferences throughout the US and Africa and in the world. In addition, we provide funding through our small grants program through Rotary clubs around the world that want to get involved in the fight to end malaria. My name is Adriana Lanting. I am the board chair. I'm from Rotary Club of Long Beach. And today we are joined by Dr. Jonathan Kayondo, who is the principal investigator for Target Malaria in Uganda. He's a brilliant man, and the work he's done is really important to eradicating malaria in the world. Dr. Kayondo, I'm going to let you tell a little bit about yourself and about Target Malaria.
Thank you very much, Adriana, and thank you for inviting me to this, forum. And I'll be pleased to, share an update about the Target Malaria, activities that we're doing, the status of what we're doing. Of course, a little bit more about myself. So I'm Dr. Jonathan Kayondo, and I'm the lead investigator for Target Malaria in Uganda. My academic background is biology, and I'm a geneticist in specialty. And of course, the work that we're doing here is to try and search for a novel tool that can be used for vector control using genetic modification approaches. And this is a tool that we hope, if we succeed in attaining it, it will be able to bring certain advantages and bridge some of the gaps that we're experiencing with the current malaria control tools. And I live in Uganda, a malaria endemic country, and I've seen malaria all around me. And I think the fight is big and huge, and it requires all stakeholders to be on board. Thank you. So now let me give you some quick overview about Target Malaria and what it is and why. So our challenge, or the reason for Target Malaria's existence, is to contribute to the fight against malaria, because malaria continues to be a serious health challenge world over, with more than hundreds of millions of infections leading to 500,000 deaths each year. And unfortunately, most of that is in Africa, affecting mainly the children, infants, and the poor. The economic losses in Africa tally to $12 billion every year. If you just have a step back, that's really lots of money. As an example, if you look at some of the infrastructure plan that the continent has, like creating some of the trans-African highways that are poised to connect lots of landlocked countries to the ports in the oceans, we estimate to need about $32 billion. And with this kind of money, if there was no malaria, we could save this in about 3 years, and then we could have state-of-the-art, transportations across the continent. So that's what we lose in terms of just money. But it's not only just money, it's also lives and lots of other things. Unfortunately, the progress in the fight against malaria has stalled, with cases rising since 2015. And so then that means something needs to be done. We can't continue as business as usual. Uganda itself is one of the top heavily most afflicted countries in terms of malaria. There we have about 95% of the population living in areas where you'd call this as a high risk areas. They're at risk of being infected. Hospital records show that up to 50% of the outpatient visits come complaining of malaria. And if you look at the hospital admissions, up to even 20%, can be attributed, to malaria. And so we need to do more to get the job done. Luckily for us, the biology has been well described of the mosquitoes and the parasites, and so the transmission is pretty well understood. And that can help us, sort of design some tools and forge some control measures if we could. So we know that malaria is caused by a parasite called Plasmodium, and this Plasmodium spread to people through the bite of infected female mosquitoes. And we know in Africa, which mosquitoes are mostly responsible for the transmission of malaria. And these are 3 closely related species that belong to the species complex called Anopheles gambiae. Of course, there are thousands, 300, 500,000, species of mosquitoes world over. About 800 of them are in Africa, but all of them don't transmit malaria. We do know the ones that do. And we also know that only the female bite and transmit the parasite. And so this forms a basis for designing, control tools. So when I speak about need for control tools, it's important people to remember that we're not saying that there are really no tools available, but the issue is eliminating malaria is the ultimate goal for most control measures and prevention efforts. And we have scaled up the currently available tools over the years, and this has remarkably reduced mortalities and morbidity. But unfortunately, this is not sufficient, and the tools have to come up faced with various complications that affect their efficacy. Among those are the emergence of insecticide resistance in the mainstay drugs, in the mainstay chemicals that are used in the nets and also for spraying, and then also in the drugs which are applied when you're sick with the disease. And there's also behavioral changes that try to make the mosquitoes avoid some of the tools that we use, such as net, so that they start biting outdoors or biting at periods when one is not under net. So those are challenges that one faces when you're in the field, when you're fighting malaria. That then means additional and more cost-effective and sustainable tools will be needed if we are to eradicate, the disease. Genetic modification has been thought as a potential tool that could combat the disease, in novel ways. And of course, WHO and even AU-NIPAD, that's the African body on development, advocates for science and technology and research into these features for genetic modification to see whether they could help with improving health and also prosperity of the people. So Target Malaria, that's how we come in. So we are a not-for-profit research consortium comprised of many specialists, including scientists and risk regulatory stakeholder, community engagement advisors. Our teams, come from Africa, Europe, and North America, because you need a concerted effort, to defeat the disease. We have over 200 experts, spread across, 11 institutes, and the network itself, the secretariat is based at the Imperial College, in London. Oh, I don't know. Okay, yeah. So the endemic country teams, we have projects going on in 5 countries based in East Africa and West Africa. In East Africa, there's work going on at the Uganda Virus Research Institute, which I lead. And then in West Africa, we have operations in Ghana, led at the University of Ghana in Accra. And then in Burkina Faso, led from the RSS Bobo in Diolaso. And then in Mali, at the Malaria Research and Training Center. And then also in Cape Verde, at the Institut National do Sud Public, NCP. And then the other partners are based in Europe and then the Americas. So a little bit about the Uganda Virus Research Institute. So our research remit is on infectious diseases, and we focus on viruses or viral pathogens, but then also the vectors that transmit those, and also malaria comes into the repertoire of the of the scope of work that we do there, and that's where the Target Malaria gets hosted here. So gene drive for vector control, this one is novel in the sense that it's only now that it seems it have been, it can be feasible, but for quite some time, trying to complement chemical-based vector control, with genetic modification approaches has been, under consideration. Even when you go back as early as the 1960s, this was thought about, but the problem was it was really not deemed feasible at that time due to various technological limitations. But it's only quite recently where we've solved some of these challenges, and we think that we have the tools that now we can start dreaming about, a genetic-based approach for, vector control. But the main thing that was limiting were twofold. First, the targets that could be used, even though theoretically you could say you knew what you wanted to modify, its location in the genome and how to target it precisely, remained a challenge. And the next was, even if you could modify a candidate in a mosquito lab, then how do you then spread that trait quickly through the wild population so that those desired trait can be harnessed for vector control? And that's where really gene drive comes in that fortunately, they're naturally occurring elements that can bias, their own inheritance, therefore making spreading themselves quickly through the population, possible. And those are the ones that we can then harness and then develop our tool and get some traits that can prevent the spread of malaria or the transmission of it. So we just harness gene drive for vector control applications, and we use, the gene drive as a tool or a vehicle. So to clarify what gene drive really is, and so that we are quite clear, because there's a slight subtle distinction between gene drive and genetic modification. Of course, gene drive is genetic modification, but not all genetic modifications are gene drive. And sometimes people mix this up. So let me say that in terms of inheritance, that's what really gene drive has. Well, that's what gene drive really improves. So if you think about Mendelian inheritance, that's the natural type of inheritance for organisms that we say are diploid, like people and even mosquitoes who carry 2 chromosomes or 2 copies of a gene, one acquired from one of their parents. So for them, if you imagine a mosquito that's altered, but without a gene drive, so it's modified. And if it's heterozygote, it has 2 copies, one altered, one not altered, but it has the trait. So if it mated with a wild type, the one that's not altered, there's a 50% chance of the offspring of that mating acquiring the modified trait, and so forth. But over generation, if this happens like this, it's not going to increase and the frequency is going to remain more or less, the way it started, unless if that trait had some, fitness advantage, or if it was helpful to the mosquito, that's when it can spread. But you can't hope to spread your trait quickly enough in, reasonably short time frame to start harnessing the benefits, relying on Mendelian inheritance. So that's where gene drive comes in, because then gene drives biases the rate of inheritance, or you can call it preferential inheritance. So then now, when you have a mating of such a modification, heterozygote, with say half of its genes, modified, the other one's not, and it mates with a wild type, now the odds of transmitting that modification is really increased beyond 50%. And now most of the offsprings that come out of that have the drive, and you can then imagine that after a relatively short period of a few generations, then this trait can spread. And that's what you really want. So then you have to attach the trait that you're interested in for vector control to a driving element, and then that spreads your trait for you. And of course, there are many traits that can be, considered or can be used to control mosquitoes. And those traits, of course, have to do with those that, facilitate, the ability to transmit the disease. And one way one could envisage doing that is to affect, let's say, the reproduction or the ability to reproduce, let's say, normally. So you could have a scenario where you're modifying, let's say, to suppress the numbers of mosquitoes, and there can be different flavors of doing that. For us, we have 2 approaches. We can do that via biasing the sex ratios to most females. If you did that, that then means in every generation, you're getting more males at the expense of females, and with reduced females, I told you it's the females that spread the disease, then over time you can break the transmission. But if this is driving, then this can happen much quicker. And the other way is to just induce female infertility directly. You target those genes that have to do, mosquito fertility, and if you deactivate them, the mosquitoes become less fertile, and if that trait spreads, then the same effect is the same, fewer numbers over a number of generations, and then you can stop malaria using the gene drive approach. So that's what it is. So I said this is a natural process in itself, that in nature you have actually driving elements out there, and those are the ones that inform this work. And a number of them have been described. You hear about transposable elements. These copy themselves in regions of the genomes where they're not, and so they increase their numbers in the genome. You have chromosome distortion mechanisms. These have been described mostly in insects, and what they do is they try to destroy the opposition or the competing chromosomes, so which don't carry the genes that they're on. And then that means that during, let's say, gamete formation, it's only those gametes that carry the particular chromosome that's distorting that gets transmitted to the next generation. So that spreads, the frequency of those traits in nature. And then there are endonucleases, a number of them, and some of them are homing. So they spread themselves via a homing reaction onto the site of a chromosome, where they're not. And so those are used as a basis to design this synthetic gene drive that we use to tag on or couple a modification or a trait that we think can be useful in vector control, in our case, to suppress the numbers, because the target malaria approach is population suppression. So we think that these genetic approaches have features that are novel and that can bridge some of the gaps that we see. For example, they have some aspect being more long-term and safe, long-term, and in that they are compatible with others and they can help bridge, gaps which others live. For example, if you thought about, let's say, resistance to insecticide, if that was the problem, then the gene drive is at another angle. So the insecticide resistance will not affect that. So you can still be controlling even populations that are resistant to insecticide if you have something else driving that controls using other traits. And then, so they're compatible with others. So they don't really interfere with others. And then, of course, they can be more sustainable because, as you can imagine, once you launch this drive using the mosquitoes, then you rely on the mosquitoes themselves, spreading this trait for you and controlling the population as the trait spreads. So then that means that the mosquitoes are the ones who are finding their fellow mosquitoes and controlling them. So you may have a break now from requiring maybe very strong health systems and cold chains if you're talking about vaccines, and then you can use them in areas which are stable or even unstable. Those gaps, because sometimes malaria control falters or is compromised when you have, say, political upheavals and those kind of things, because the mainstream methods require a good logistics network, which may not always be possible. And then they're more cost effective in that because it's a drive, you don't have to start with lots of numbers or many billions of them. You can start and seed with few, and then they'll spread the traits themselves as the reproduction goes on. And you don't need to have behavioral change among the population. Sometimes if you give people medicine, you must make sure they are adherent and taking their pills as prescribed. Or if you give them nets, then they have to sleep under them. And sometimes they don't, they use them for other things. So this one kind of bridges some of those gaps. So those are the features. So our work has those objectives. I say target malaria, we are reducing the numbers of the population that transmit the disease. And as this reduces, we hope then that the malaria transmission can be capped, but we reduce through genetic modification. And we also hope to develop and share this technology widely with a number of stakeholders and those that may need to use it when it's ready. And of course, our approach is aimed at being complementary to existing methods. So it's quite compatible and it has a longer term aspect. And it's pretty targeted. Like I told you earlier, there are many thousands of species of mosquitoes, but our work is only targeted at those malaria Gambi complex species members that are the main transmitters of the disease locally. And we do this just to help save lives of the people who die from malaria every day, who are mostly children, young women, and so forth. So the technology or our research is built on 3 pillars: strong science, stakeholder engagement, and of course, adherence to regulation. And we take each of those quite seriously. And we have really dedicated teams that specialize in doing that. And in terms of current status of progress, I just want to say that at the moment, let's say globally, the technology development status is we have tried out and developed, gene drive, so non-gene drive genetically modified, mosquitoes. And this was just for capacity building and training purposes. I said there's a distinction between gene drive and modification. Sometimes you can modify a trait, but not attach a drive to it. So the version I'm talking about that we've done and tried out in some of our partner labs in Mali and Burkina Faso are those versions where they have a modification of interest, but they're not driving. So we started with a modified staromere, and we did this so that we start with the most, let's say, easiest or the simplest version or complex that we can get, and use it as a basis for training on handling genetically modified mosquitoes and build infrastructure that can do this kind of work. And of course, because they don't spread, because they're sterile, so they don't spread this modification, then they are single generation. And then once they finish their life cycle, then they're dead and they're gone. So the modification does not persist. So that work happened in Burkina Faso, where they imported in 2016 and then they did work under containment, checked a number of, assumptions and confirmed, and they got findings. But the most important thing was to be able to get the capacity and build infrastructure and test it because no genetically modified, work had happened in mosquitoes in Africa before then. And then they also went ahead and did some small scale release again to get some other assumptions in the real life and in the field. And that was in July 2019. And Mali followed suit. They did contained studies and they are wrapping up. They wrapped this up in July this year. Now, the next stage of our work is to have non-gene drive, but genetically modified male bias. So these ones will be fertile, but they are still not driving as a result of the nature of the design. And for those, there are some questions that we cannot answer using, for example, sterile versions, because if you wanted to study how the modification is transmitted, the other ones are sterile, you cannot do that. But this will allow us to do this kind of work and also check some other aspects of that in the field as well. But that's the next work for Burkina Faso and Uganda. We'll start in the lab, and then when that's done and successful, then later on, after some time, then we'll go to the field. But all that depends on whether the regulation, we pass and whether we get the findings that we expect to get before we move to the next stage. So the development is still a long way to go. And then we have teams in Ghana that are looking at the ecological impacts of the role, for example, of the mosquitoes in the ecosystem. Some people worry about that. And so they're trying to provide answers to the question, what does mosquito do in the ecosystem and what's the role of it? And we are not many clear studies on that. So we're really looking intently because that's important in some of the things that comes up when we engage with stakeholders, and also how to improve the rearing in masses of our colonies. And then there are also entomological studies in Cape Verde that are also going on. And then the gene drive version, there are a number of flavors that we could do, like I talked about. You can bias the sex ratios, or you can just induce infertility, like deactivate those kind of genes in the mosquitoes. That's still in the development and in the lab, and it's still not ready for testing yet, in the field. It's still a working process. So for Uganda itself, what we can say at this point in time, we've finished laying the biosafety, groundwork for this kind of work, because, like I said, because it's genetic modification, it's a first of its kind. You require the right infrastructure and the right containment facilities at the right or the recommended levels of containment by international good practices. And we chose a level that's recommended by a number of bodies, like the American Control of the Medical Entomology and the WHO. We recognize arthropod containment level 2 containment. And that's what we built and established. And we opened this up about 2 years ago. And we've been doing some dry run using not genetically modified mosquitoes, but normal wild type caught mosquitoes, but being reared at this high containment level to make sure that it's functional, it works, and the teams are well trained in the protocols and so forth. And that's going well. We should be wrapping this up pretty much soon. And then after that, we hope we can then go to the next phase of the work, which is trying to get permits to work with genetically modified mosquitoes, but with versions that are not driving yet. So that's the next stage of work, but we have to go through some regulatory applications and things like that. And then if we're found, qualified and not qualified, if we're found ready according to what we'll have done before, then maybe we'll be allowed to continue, but we're hopeful we shall. Now, while that was happening, we had to do some baseline entomology just to understand, the population of the locally existing mosquitoes, because those are the ones that we'll have to control using genetically modified approaches. So you always need to have a good baseline. And we looked at a number of things like the species composition, the abundance, their seasonality, and their dispersal, their biting rates, and things like that. And then we are zeroing in on potential candidate sites. Now, I said that the pillar of work is stakeholder engagement. So we've engaged lots of stakeholders because we want this to be informed and we want the work to be accepted from an informed way. And so we have the dedicated stakeholder engagement team engaging at different levels, at the national level, at the village site level, and so forth. And it's a work in progress. And it also increases according to the stage that we're at. When we're at the baseline and everything, we focus more at the study sites. But as we keep approaching the genetically modified stage, then we sort of amplify this kind of engagement and reach out to even more and more stakeholders. And then there's the regulatory oversight. That's also the third pillar, and we comply with all international, regional, and national regulations. We try to have best practices in each of those to as much as we can. And we are always aware that risks have to be assessed and managed properly and we have experts that help us with that. But we also consult at all stages. We're working with the institutional biosafety committees to oversee this kind of work, but then also at the national level, there are competent authorities that we exchange with and get guidance from. And also we have environmental aspects. Maybe lastly, I want to just say that even though I've talked about Gene Drive and the promise that they have, I don't want to leave it that now they're the magic bullet and themselves, they don't have issues. They also have, of course, issues. And one thing is they too can suffer with, complications like, the other tools do. For example, it's feasible to develop, for the mosquito to develop genetic counter resistance to even the drive. So that's something that have to be watched out. And when you design them, you always have to be worried of that. And then there's the issue of governance, regulation, and public acceptance. This is a new thing. I think people need time so that they understand clearly what's going on. And it's going to be one of those things whereby we learn as we go, but one has to start it because malaria is very important. And then the unintended ecological and biodiversity effect, or the effect on the environment that people worry about. This has caused a number of reactions. Some have said, let's have a research moratorium until we understand everything. But then others say, no, I think there's a way. We could have a step-by-step or slow development pathway which allows us to learn as we go without doing full scale. And the Target Malaria team subscribe to this approach. I would like to end it there and acknowledge our funders, the Bill and Melinda Gates Foundation and the Open Philanthropy Project, but then also our PIs get other funding from other bodies as well. So malaria is big, probably no more than one funder can manage it all by themselves, and it's a collaborative effort even at the funding level. And these are the logos of the partner teams. And thank you very much.
Thank you, Dr. Kayondo. Wow, I'm surprised. There's a lot of things that I was surprised to learn there. So, and I'm sure everybody else feels the same way. For example, 3,500 species of mosquitoes. I had no idea there were that many.